Let's talk about amino acid structures. So what is an amino acid? You've been talking about these probably in your intro bio classes. It's not the first time you've seen it, but I want to talk about it really from a biochemical focus and start with that definition. So an amino acid is a compound that contains both an amino group and a carboxyl group. So looking here, we can see our amino group, our NH3+, and our carboxyl group, COO-. And then we see we've got this alpha carbon, C alpha, the alpha carbon in the center. We've got a hydrogen, and then we've got the R group. So the R group indicates the amino acid side chain, which is going to be different for each of the 20 amino acids. Amino acids can theoretically exist in two different stereoisomers, L and V. So do you remember stereoisomers from organic chemistry, where two molecules can be mirror images of each other? Kind of like your hands. If you put your hands out in front of you, you see that your hands are non-superimposable mirror images. So in chemistry, molecules can be the same thing. Those are the two stereoisomers. So you can see the L form on the left here and the D form on the right. Only the L form is commonly found in proteins. The D form has some special cases. You see it here and there, but primarily for human biology and biochemistry, we're going to be talking about the L form of amino acids. Most amino acids exist as Zwitter ions at neutral pHs. So again, I'm talking about free amino acids, not bound into a protein, but when they're free in solution. So remember that a Zwitter ion is an overall neutral molecule that has a sum of charges that add up to 0. So for example, if one part of the molecule has a -1 charge and another part of the molecule has a +1 charge, the total sum is 0, but it does have charges inside of it. So for example, the Zwitter ionic form of our amino acids, shown in this blue trace, is what we see here, where we've got our positively charged amine, our negatively charged carboxyl, and then overall the charge is neutral, which makes this free amino acid a Zwitter ion, assuming, of course, that the R group is also neutral. As we move to a more acidic pH, we can protonate this carboxyl, and then we'll have an overall +1 charge on this amino acid. And as we move to a more basic pH, we can deprotonate the amine, so now we just have a negatively charged group. There are 20 common amino acids. There are some uncommon amino acids that gets us past 20. We're not going to really cover those in this class, but just so you know they exist. We're going to focus on the 20 common ones. One way we can sort these is into essential and non-essential amino acids, where essential amino acids must be consumed from our diet. Whereas in contrast, non-essential amino acids can be made metabolically. So if I have the components I need to make those, my cells can make those non-essential ones. Whereas the essential ones, I have to get them from some food that I ate. That's not going to come up too much in this biochem class, but for those of you who are more nutrition focused, it's an interesting component of human nutrition. All right, so here is the 20 common amino acids. For each one of these, let's take alanine, for example. You can see the amine, you can see the carboxyl, the hydrogen, and the alpha carbon. And then up on top, these are all arranged the same way. You have the R group. So you see the R group is different for each amino acid. So for alanine, it's just a methyl, CH3. For cysteine, it's that carbon and then sulfhydryl group. For aspartate, we have that carboxyl over here and a methyl. So this whole section right here that I'm circling, that is the R group for aspartate. You also see that each amino acid has the full name, a 3-letter code, and a 1-letter code. So I've organized these amino acids for you in alphabetical order, which is probably the least useful order to have them in. So what I would like you to do is practice sorting them based on the chemical properties of their side chains. So we're going to do this in class, but you're welcome to get started on your own. Sort the 20 common amino acids into the following categories based on their side chains. So the aliphatic, nonpolar, the aromatic, nonpolar, the polar ones, excluding the charged ones, the positively charged, which are also referred to as cationic or basic amino acids, and the negatively charged, or the anionic and acidic amino acids. So again, take a look at just the side chain. Don't consider the backbone. Don't think about the backbone, but looking at just the side chain, could you sort these 20 amino acids into those 5 different categories? And then for each amino acid, write the full name, the 1-letter code, and the 3-letter code. And you might as well redraw it for yourself as well. So what I would do is take a blank piece of paper or maybe a new page on your tablet, whatever you're using to take notes. Set up these 5 categories and then just start placing amino acids into them, drawing the structure, writing the name, 1-letter code, and 3-letter code.